So today we are going to finish up with the biotechnology lessons. We will definitely have time to do that. And then next week we will do the final lecture, lecture 10, which serve as a nice transition connecting point between this course and the one that you will be taking in the second semester. So at the end of last class, we were talking about the plasmid. So let me just recap some of the key points that you should know from last class first. Right, so this is class notes, November 30th, 201. All right, so what do we know so far? What do we know so far? We are trying to clone a gene of interest and put it into a plasmid, and then we're going to feed the plasmid into the bacteria such that the bacteria is going to mass produce the protein that is encoded within the gene, right? So this is what we have done so far. You're gonna have your DNA, which is copied or amplified, we say. Amplified by PCR. Right? DNA, which is containing the gene of interest. Okay? In our example, the gene of interest was a gene that makes insulin. Okay? So you clone that, you make billions of copies of it, and now is in this, tiny tube. And what we're going to do is we are going to be basically treating this with a little bit of restriction enzyme. Restriction enzyme. Okay, and what that does is, let me try to draw this again. What that does is we're going to have a gene, and the restriction enzyme that we use are going to create sticky ends at the flanking ends. Okay, so the restriction enzyme creates sticky ends. Okay, but we must make sure that the restriction enzyme is not going to be cutting our gene. And the way to do that is to make sure that the recognition site of the enzyme does not exist inside your gene. So let's say the enzyme is going to cut AAA-TTT. So we have to make sure that the AAA-TTT only exists at the ends of the gene and not within the gene. Okay, otherwise you destroy the gene and there's nothing we can nothing we can use. All right, on the other hand, we are going to have a tube of plasmid. Okay, so this one is gonna have the gene in it. Let me just use a maybe like green color or something. So that's gonna have the gene, and then this one is gonna have the little plasmid. I mean, you obviously cannot see them; they are microscopic. Okay, but as a picture, that's what it would look like. So these are plasmid. Okay, and then we are going to be using the same restriction enzyme to cut the plasmid. Okay, must be the same. Same enzyme to create compatible sticky ends. All right. So there is going to be your plasmid. And we're going to be cutting it open. And you will have the sticky ends. like that. Okay. So again, sticky ends. And let me just color the gene green. Like that. And then you're going to mix these two things together, right? And you're going to add a little bit of ligase, right? Plus ligase. Then you're going to end up having the plasmid with the gene of interest in it. So this is, let me try that one more time. There we go. So this is a plasmid plus gene of interest. Okay, and all this is going to be sitting in a tube, sitting in like a tiny tube. It's really small. It's no bigger than this. Okay, and you're going to have like billions of these things in it. All right. So I guess like you will have the little plasmid, but these plasmid are now going to have like little green bits in it. I mean, you don't really have to draw like that, but just for you to visualize. Okay. Then we are going to feed this to the bacteria, right? We're going to feed this to the bacteria. Transform into bacteria. Okay? Transform means you make them take it up, basically. Okay, so like you would have another tube of bacteria, and then you have the plasmid, and then you would mix them together. So in the end, we're gonna have the bacteria. And these are bacteria. Okay, and then you know some will take it up the plasmid. Okay, maybe some will not. Okay, so this one accepted. This we'll say these ones, they are transformed. Okay, they pick up the plasmid. And we can use different methods to encourage them to take it. We can heat them up. We can give them some cold shock. We can give them some, you know, electric current, different ways to encourage them to take it up. And then these bacteria, these ones here, they did not take up plasmid. All right. But you can tell just by looking at it, they look the same. All right. And then you're gonna grow them on a plate, and then you're gonna see whether they grow or not. Okay. So to distinguish which bacteria have accepted, have been transformed in. You can say transform means accepted the plasmid. We need a selection method. Okay, we need a selection method. So let me just copy this plasmid over here. We talked about this already briefly last time, but let's you know revisit the idea. Okay, so here is the plasmid that we buy online. This is just the name of the plasmid. Name of the plasmid. Okay, no particular meaning. This is the origin of replication. That's where you start the process of transcription, right? Basically. And all these names on the outside of the plasmid, those are the it shows where each restriction enzyme can cut. Okay. In other words, these are insertion sites for genes. Okay. For our gene. You know, the green thing we're trying to put in, right? So like, if remember, let's just go back here for a second. In order for us to insert the gene into the plasmid, we have to cut them with the same restriction enzyme, right? So if I cut this with, if I cut our gene with E-core-R1, which is the restriction enzyme, then I have to cut the plasmid also with E-core-R1, and then your gene is gonna go in right here. Okay, you're gonna be inserting the gene like right here. However, if you treat your gene with PST1, then you must also treat the plasmid with PST1. In that case, the gene is gonna go right here. Okay, you cannot treat your gene with E-core-R1 and treat your plasmid with PST1 and then try to put them together. That's not going to work because the sticky ends that are created by the E-core-R1 is not going to be compatible with the sticky ends that are created by PST1. Okay, remember these sticky ends are actually nucleotides, right? So like if this one is like AATT, you need to have a TTAA to be compatible. And that's only possible if you are treating it with the same restriction enzyme. Does anybody have any question so far? Do we all understand what the yellow sites are? So why would you be choosing E-core-R1 over PST1, for example, right? Well, like I said, when you treat the gene that we have with the restriction enzyme, we have to make sure that it doesn't cut within our gene, right? And sometimes, because we don't control the nucleotides of the gene, right? The ACTGs of the gene it is what it is. Like we cannot alter that. So like again, theoretically, if your restriction enzyme cuts AATT and then there is AATT within the gene, you cannot use this particular restriction enzyme. So you have to try another one, right? So that's why you need to have multiple options when it comes to what kind of restriction enzyme we can use to insert the gene into the plasmid. Okay? Now, these other stuff that are inside the plasmid, the names, okay, these are selection markers. Selection gene, selection markers. Okay? We can use them to help us distinguish which bacteria have been transformed. Okay, it would give them some special properties or a special appearance in some cases if they have accepted the plasmid. Right? So in this case, these are antibiotic resistant genes. Okay, so the AMP here, that's ampicillin resistance. And then this is tetracycline resistance. These are just like different antibiotics, right? Okay. So let's take a look at what happens. If you have a mixture of bacteria, right? I'm just gonna copy it from, you know what, doesn't matter. Let's just draw it again. Okay, so you have a bunch of bacteria, right? Okay. And let's just say this is non-transformed. do not have the plasmid. Okay, do not have the plasmid. Okay, and then you're gonna grow them. You're gonna put them on a plate. Okay, you're gonna put them on a plate. It's like a little dish with nutrients. Okay, so just basic nutrients. Now, let me show you like a plate. Maybe you don't know what I'm talking about here. I'm just trying to copy a picture over to show you. Copy image. This is a agar play. AGAR agar play. And basically it's like nutrients and like give bacteria a place to stick on, and then they will grow. Okay. And then I'll show you what it looks like when they grow. Hold on a second. There. So like they grow, right? That's like bacteria growing. Okay. So for them to grow, we need to give them like nutrients and stuff, right? So this is just like a plate with basic nutrients. And these are bacteria that have not accepted the plasmid, and then I'm just gonna put them in here. Do you think they're gonna grow, or do you think they're not gonna grow? Grow or not grow? Well, this is a pretty basic. Hopefully, you will agree that they're gonna grow. Right? They will grow. Right? They will grow these big colonies. Okay, everywhere. Okay, everywhere. Let's make this bigger. Right? Okay, so they will grow. Right? Billions and billions of bacteria. Okay, so we call this lawn growth. Okay, lawn growth. It's like they grow so much they cover the whole lawn. That's basically what they're saying. Okay. Now, what about this? What about I take these bacteria and then I put it on another plate. Okay. This time it is basic nutrients plus ampicillin. Okay, the antibiotic. They don't have the plasmid. They just like regular bacteria that have rejected the plasmid essentially. Do you think they will grow in the presence of ampicillin, the antibiotic? Yes or no? Come on, you can do it. Tell me, will they die or not die? Will they die in the presence of ampicillin, the antibiotic? All right. If you don't want to talk, that's okay. Let me launch the poll. So A means they will grow. B, they will die. What do you think? Okay. Only a few of you have made a choice. That's okay. So they will die. Okay, they will die. No growth. Why? Because the ampicillin will kill them all, right? Ampicillin will kill the bacteria. Okay, will kill the bacteria. All right. Let's take a look at the other bacteria right here. We have another group. Let's say they have accepted the plasmid. Okay, they have accepted the plasmid. These are transformed bacteria. Okay, see, which means they have accepted the plasmid. Okay. Now, in the plasmid, there is the ampicillin-resistant gene. Ampicillin-resistant gene. So what happens is, now, if you put them into, let's put the plasmid in here to show that they have accepted it. Now, if you put it into the plate, right, that has the basic nutrient plus the ampicillin, they will still grow. You will see growth. Okay. Why? Because the plasmid has, or I should say the plasmid gives them resistance to ampicillin. Okay. It gives them the ability to grow in the presence of ampicillin. That's basically what's going on. Okay, so that's how they will behave differently if they don't have the plasmid or if they have the plasmid. Okay, so this is growth. You will see, growth. Okay, no growth here. Now, if you're still with me, let's go back here to our experiment, right? So over here, this tube contains bacteria that are transformed as well as bacteria that did not take up the plasmid, right? It's a mix of them, right? So what happens if you take this and put it in to the plates? Okay, so this is just nutrient. This is nutrient plus, let's say, ampicillin. Okay. Well, if it's just nutrient, you would expect them to grow. Whether you have the plasmid or not have the plasmid, it doesn't matter. They are going to grow, right? But if it's nutrient plus ampicillin, you will only have some growth. Some growth. Okay? Only transformed bacteria will survive. And those are the ones we want. We want the one that have accepted the plasmid. Those are the one that's going to be producing the insulin for us, right? So that's how you can tell whether they have accepted it or not. Okay, and this is just basically like a control for the experiment, right? That's what happens if if you don't have antibiotic, they're supposed to grow. But once we add the ampicillin, you would expect reduced growth. All right, let's take a look at the PowerPoint again. Here is a question. A gene is inserted. You know what? Let me just copy this over to the notes so we can take a closer look at it. Oops. A gene is inserted into a plasmid using E-core R1. Okay, so E-core R1 is right here. The plasmid is transformed into a tube containing E. coli. You cultured all the E. coli on a plate containing tetracycline, which is an antibiotic, What result do you expect? Okay. So if I insert a gene using equal R1, then the gene is going to go right here. It's going to go right here. That's our gene of interest, right? Because that's the insertion site for equal R1 according to the plasmid map, right? It's going to go right here. The green is the gene of interest. Okay. And then you transform it to a tube containing E. coli. Right? Not all of the bacteria will accept the plasmid, right? Some will not. You cannot guarantee 100% successful transformation. And then you're going to culture it in the presence of tetracycline. Well, this plasmid has a tetracycline-resistant gene. So if the bacteria accepted the plasmid, they will be able to grow. Right? They will be able to grow. If they don't accept it, they will not grow. So what's going to happen? All of it will die? Well, no, right? Because some will accept. Some will accept the plasmid. All of it will grow? No, that's not true. Some will not accept the plasmid. Okay, if the bacteria accepted the plasmid, they will grow. They will have tetracycline resistance, so the antibiotic's not going to bother them. If they do not accept the plasmid, then the antibiotic will kill them. So, at the end, in the tube, there is a mix of both, and the answer is only a small portion will grow. Okay, only a small portion will grow. Let's take a look at the second question here. It's the same question with one difference. Okay, same question with one difference. A gene is inserted into the plasmid using a different restriction enzyme. Okay, this time we're using SMA1, small one, and everything else is the same in the question. Same plasmid, same conditions. Do you think they will grow, or do you think they will die, or like only some will grow? What's going on? What do you think? Okay. Thank you for those who have participated. The correct answer is that they will all die. They will all die. Here is why. We are inserting using SMA1, which means the gene is going to go right here, right? The map tells you that that's the insertion site if you use SMA1, right? Inserting a gene using SMA1 will basically disrupt the tetracycline resistant gene, right? You're inserting your gene in the middle of the tetracycline resistant gene. You've broken the tetracycline gene. Okay, it's not functional. So even if the bacteria accepted the plasmid, they are not going to have the tetracycline resistance because the gene has been destroyed. The tetracycline resistant gene has been destroyed because we inserted something in the middle of it, right? So in this case, no matter whether they accept or not accept, they will all get wiped out by the tetracycline. Now, I don't know if you guys have been doing the practice quizzes on online, but because chapter 9 is kind of difficult, you definitely should try them out. Okay, so if you go to lecture 9, right, biotechnology, uh, there are like the self quiz, right? Okay, and you can uh, I mean on yours it will say start the quiz like this. So there are some good questions here that you should try out, right? So look, there are plasma questions. These questions are very similar to what you will be seeing. Okay, so at least try them out. Make sure you have some good understanding of it. Okay, I'm just gonna turn that off. Done. So any additional question before I teach you the next key concept? So that's basically how you use recombinant technology to tell the bacteria to make insulin, right? It's recombinant because the plasmid originates from, the bacteria, and the gene is the insulin gene from human. So you put it in, and now it's a combination of two sources. And then you feed this into the bacteria. Once they accepted the plasmid, they will mass produce whatever product is encoded by the gene, right? And then you just need to have a system to extract the insulin from the solution. Well, if you guys don't have any question, we're gonna move on to um, the next concept. Okay. So another biotechnology we're gonna look at is DNA fingerprinting. You might have heard of this. DNA fingerprinting is a method to match DNA samples with individuals. So let's say someone found some DNA in a crime scene and they want to determine, who it belongs to, and then there are a few suspects. You can compare the DNA in the sample with the DNA of the person, and it's very, very accurate, up to 99.9% accurate. It can also be used to determine if individuals are related, right? If there are concerns about, who the father is, for example, and, you can test it out. Test the samples from the man, and then test the samples from the child, and DNA fingerprinting will tell you with certainty, who's related to who. So how does that work? Well, we're gonna spend some time talking about it. But first, there's this new term. It's called polymorphism. Polymorphism. Polymorphism is any difference in DNA sequence that can be detected between individuals. Okay, so we talked about previously how even though humans are 99% identical, like any two individuals are 99.9% identical on the genetic level, there are some differences, right, among us. And those differences, if we can have a method of detecting it, right, that difference is called polymorphism. It can happen in the coding regions of the DNA, which of course are the exons, as well as the non-coding regions. Okay, there are some fillers, right, in our genetic material that either they don't do anything, or like we haven't discovered what their functions are just yet. Okay, just like in the textbook, right? Like if you read a whole textbook, there are some stuff that, you feel like it doesn't really need to be there. It doesn't really help you understand anything, like maybe like an irrelevant picture or something like that. So the non-coding regions are kind of like that, right? They're like filler stuff that seemingly doesn't really do anything. Right? So the polymorphism, the differences can occur in those regions as well as the coding regions. Now, within the non-coding regions, one type of polymorphism occurs in what's called short tandem repeats, or STR for short. So what is an STR? An STR is a bunch of repeats of the same nucleotides over and over again. Short as in, couple nucleotides long, 3, 4, right? Sometimes it could even be 2. And tandem is one after the other. And then they are repeating over and over again. So for example, here we have CAG, 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 CAG. So we call this CAG4. It's repeated 4 times. Okay. So for me, I might have this STR on chromosome 17, and then it's repeated 4 times. If I look at the exact same location on your DNA, chromosome 17, and I look for the CAG, right? Since we're not related, the number of repeats are likely to be different. Okay, people who are related tend to have the same kind of repeats, right? So if I'm repeated 4 times, maybe on yours it's repeated 6 times. Okay? So I told you before, when it comes to these repeats, it's very easy for your body to make a mistake when they're copying it. Okay? Because they miscount the DNA polymerase when it copies the CAG. Like if it repeats like 10 times or 20 times, it's easy for it to maybe make an extra one, or like maybe they copy one less, right? And that's what's responsible for creating the polymorphism in the human population. So one disease called the Huntington disease is related to these tandem repeats. Okay, so Huntington disease is a progressive brain disorder that causes uncontrolled movement and ultimately will cause the loss of cognitive capabilities. Like think it's going to affect how the person's mind, basically. Okay, they will not be able to think on their own properly, right? And if you're thinking uncontrolled movement, isn't that like, Parkinson disease, and then, loss of cognitive ability, isn't that like Alzheimer's disease, right? Actually, Huntington disease, Parkinson's disease, and Alzheimer's disease, they are all neurodegenerative diseases. Okay, it causes your brain to slowly break down in general. Okay, so they have identified a gene called the HTT gene, and within this gene, there is a CAG segment. And in normal individuals, as in people who don't have Huntington disease, the repeats is about 10 to 35 times. Okay, so if it's CAG 15, it means it's CAG, 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 CAG repeated 15 times. Okay, so like for anybody who doesn't have Huntington disease, it's between 10 to 35 times. However, people with Huntington disease, the CAG segment is repeated 36 to 120 times. Right? So there seems to be a threshold, a boundary, a limit, okay, a limit beyond which the disease starts to show up. Okay, and CAG codes for glutamic acid, right? If you look it up on the codon chart, right? Okay. So, people who have Huntington disease is actually a dominant condition. Okay? But the symptoms does not actually show up until after age 40-ish, generally speaking. So by that time, people who wanted to have kids, most of them would have had kids already, right? And let's say the person is big H, small h, and then they have kids with someone who is small h, small h. So if you do the Punnett square, right, you will see that there is a 50% chance that their child is going to have the Huntington, right? So it's really scary for some people if they You know, if they found out their father has Huntington, they could get tested. They could go do a genetic test to see, you know, if they also have the tandem repeats that are between 36 to 120. It's an easy genetic test. Uh, and then they would find out, right? So there's this dilemma, right? Do I find out or do I just not find out? Right? Some people will think, you know, finding out, at least I can prepare for it, and then, you know, I'm not gonna have a family because I don't want to pass it on to the next generation. Other people doesn't want that dark cloud hanging over them, right? They don't want to deal with it, you know, just live life to the fullest. You know, so there are some difficult choices, right, in those situations. Now, it's also possible for a family to have no history of Huntington, and then it shows up in a generation out of nowhere. Okay, so one possible explanation for that is, let's say for whatever reason, the CAG repeat is long for this family. Okay, not so long that it's gonna trigger the Huntington, but you know, relatively long. Let's say it's like, you know, 33 copies, okay, or even 30 copies. And I was telling you, when you have these repeats, it's easy to make mistakes when you're copying the DNA, right? When the DNA polymerase is doing the copy. So let's say in meiosis, the person is creating the gametes. That also involves DNA replication, right? So let's say, you know, many generations back, it started as like 30 repeats, but then the polymerase made a mistake. It slipped. It made an extra few copies. So 30 becomes, say, 33. Okay, and then it was okay for a couple of generations, and then maybe another mistake happened, right? Somewhere down the line. And then from 33, it grew to 37. Then all of a sudden, you cross that threshold, right? You cross that boundary, and then the Huntington disease might showed up. Okay, so that's a example of like what the tandem repeats are. But that's not why we're talking about it. We're talking about it because we want to use this STR situation to look at DNA fingerprinting. Okay. So the short tandem repeats are unique to every person. Yeah, and that's why we can call it like DNA fingerprinting is as unique as your fingerprint. So for person one, they might have like an AGAT repeated 12 times. Someone else might be repeated 5 times. For some locations, it could be the same. Okay, that's why we don't just look at 2 STRs or 3 STRs. We look at as many STRs as we have discovered, and we compare all of it. All right. So I'll come back to this concept later. But first, I want to introduce you to this technique called gel electrophoresis. Okay, gel electrophoresis. The purpose of gel electrophoresis is to separate DNA based on their size. So I'm gonna keep things simple for you. All right, we're gonna have this chamber, and you're gonna fill it up with a buffer solution. The buffer is just helps conduct electricity. Okay. You know what? I think I'm just gonna copy this over to the OneNote here. It's just easier for you to see afterwards, right? I suppose. So this buffer is to conduct electric current. Okay. Now we have an electrode here that's negative, and this is positive, and then you hook it up to a power supply, and basically the current is gonna be going from negative to positive like this. Okay, you will see the solution bubbling as the electricity is going through it. Okay, and if you think about the DNA backbone is negative because of the phosphate, right? It's a sugar phosphate backbone. That's one of the first things I taught you in the course. So if you put the DNA here in the gel, and I'll tell you more about the gel in a bit. If you put the DNA here, that's the DNA, the blue stuff. The current, which is going from negative to positive is going to push the DNA towards the positive electron. Okay, so the current pushes the DNA towards the positive terminal. Okay, towards the positive terminal. So let's talk more about the gel. Okay, the gel. This gel, it comes in powder, kind of like, milk powder, baby powder. What do you call that? Baby formula, right? Like those powders. And then you just dissolve it in some solutions, and then you microwave it, and then it's forget the baby powder. It's more like making Jello. Okay, Jello comes in little powder format pouches, right, as well, right? So you put it into the liquid, and then you microwave it, and then you can pour it out into a mold, and then you just let it sit. Okay, and then just like Jello, that will turn into solid eventually. This gel becomes solid. Okay, so the gel looks like this. Okay, the gel, if you zoom in, it's like this microscopic mishmash. Okay, of a mesh, if you would. Okay, the more powder you use, the tighter the mesh is going to be. Think of it as like like a strainer, right? That you might use for cooking, right? Like if you want to drain the liquid from your pasta or something, then the strainer has like really really big holes, right? Okay, but like if you want a strainer to clean your rice or something, then the mishmash has to be much tighter, right? Do you guys understand what I'm trying to explain here? I'm sorry, the picture is really bad. Okay, but the mishmash, right? It could be really tight, like here, really tight, or it could be really loose, and you can control it. You can calculate that, right? And see how much powder you're gonna use. Right? So the current is pushing the DNA through the gel, through these mishmash. All right, let's just say this is the beginning. Okay, this is the beginning. This is where the negative is, and this is where the positive is. And then I have DNA. I have DNA that is really, really long, and I have DNA that is short. If it's really short, the current is going to push it, and it's going to be able to weave itself through this network really easily, right? Whereas if it's really, really long, it's still going to go, but then it gets tangled up easily, and then it will just stop moving altogether. Okay, it's going to take it a very long time to pass through, right? So the gel is going to be able to separate the DNA based on how long it is. The longer it is, the less distant it's going to move. Shorter ones, they can go very, very far down. Okay, so let's say we have a gel here. Let's draw the gel. Here's the gel, and then we have some slots. We call them wells, actually, some wells to put the DNA in. So load DNA into well. Okay. You're also going to add a blue dye to help DNA sink into the gel. Okay. And also gives you like a visual way to track the approximate location of the DNA. Okay, so you're gonna load your sample into the well, and then you're gonna apply the electric current. Okay, the electric current. This is negative, this is positive. It's gonna push the DNA forward, and then they will separate. So let's say you separate the DNA like this. Okay, separated DNA. So the further it moves down, the shorter it is. So this is the shortest DNA. And then over here, this is the longest DNA. Okay. So like if you zoom in to look at the DNA in here, they would all be really, 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 really tiny. Okay. If you zoom in here to look at the DNA, they would all be very, very long. Questions about this technique? All right, let me show you a video of how it is actually done, and maybe you will understand it a little bit better uh, once you see it. So I'm gonna have to pause the recording here because otherwise they won't let me post it on YouTube afterwards. Yeah. I do have an analogy for you, like to understand this. I told my kids to the park to like an indoor playground one time, and there was this play structure. I guess it looks like this. I don't know if you guys have seen it before. So it's this really tall. It's like two-story tall tube basically, and then in the tube there are like different levels. You get different levels, and each of these rings have a lot of tapes in it. Okay, like really really long tapes. Okay, like that. All these tapes. So the tapes are really strong. Okay, like if you jump in from here, you're not gonna fall all the way through and hurt yourself. Okay, but and the fun of it, I suppose, for the kids is they have to kind of weave themselves through these tapes, and either go down or go up. Right, depends what they want to do. Okay? So my daughter was like, hey, you know what? I want to race you to the bottom. Okay, let's get in together at the top here, and then we'll race down to the bottom. Okay, I was like, yeah, sure, let's do that. Right. Okay. But because she was really, she's like, like a kid or like a 5 year old, so she's like tiny compared to me. She was able to weave through this thing very quickly. Just like the DNA, if it's short, it's able to go through the mishmash easily. Me, on the other hand, I get stuck. Right? I literally got stuck in the first one, and then it took me a long time to get to the bottom. And that's important to remember. It's not that I cannot get to the bottom. It's just that it's gonna take me some time because I'm big and I'm gonna get like stuck here and there, right? And that's why when we did the gel, when you saw the video, right? If you leave it on for too long, eventually everything will be lost on the other hand. Even the long ones is gonna be able to make it through. Right? So that's why we have to turn the machine on, but not forever, just for like some time, and then we will stop it so that we don't lose the sample. Okay? So I'm gonna pause here. Let's take our break, and when we come back, I'll tell you how we can combine our understanding of STR with the technique of gel electrophoresis to do DNA fingerprinting. Okay? So just 5 minute break. We'll resume. See you soon. All right. Let's put everything together. Okay? This is gonna be a big note. Okay. So make sure you give yourself plenty of space. So let's say we consider two individuals. Okay, so this is called STR analysis. Okay, using gel electrophoresis. Okay. So let's say we have two people. Right? Two people. I call him Bob or something. And then we're gonna have another person here. Sally. So let's take a look at Bob here. Right? Bob, we're gonna examine his chromosome. This is chromosome 2. And, let's say there is an STR here on chromosome 2. This is CG repeated 10 times. So how long is this in total? If it's CCG repeated 10 times, how long is it going to be? Okay. 3 times 10 is 30. So that's 30 base pair long. Okay. 30 base pair. And then we're going to look at another chromosome of his. Okay, this is chromosome, let's say 14, which is why it's shorter. And then let's say he has another, there's another, hold on a second, the pen is not really working. Okay, let me turn off the video. I think it's that's what's slowing everything down here. You don't need to see me. Okay, so let's try again. Okay, chromosome 14. We have another thing here. Let's say this is AT, and then repeat it 3 times. So that's 2 times 3, which is 6 base pair. And then let's just say we do one more. Let's just say we look at chromosome 21. And there's one last one here. It's a TTC. TTC, and this one is repeated 20 times. Okay, so that would be 6T base pair, right? 6T base pair. So we're gonna look at Sally as well at the exact same location. Okay. Everybody has the same STR in the same location. It's just that, the number of repeats might be different. So for Sally, maybe, CCG, and it is also 10 for her. It just happens to be the same. They don't necessarily have to be related. It just happens to be the same. And then, for chromosome 14, we're gonna have another repeat here, AT, which is, let's say, repeated 10 times. So that would be 20 base pairs. And then finally, chromosome 21, there is the TTC again, but this time it is repeated, let's say 5 times. So 15 base pairs. Okay. Okay, so I'll give you a moment to copy this down. You gotta do this in color, otherwise the note will not make sense. So just to quickly recap, in case you don't quite fully understand it the first time, we are just looking at these short tandem repeats at different locations in the genome for 2 people, Bob and Sally. Okay. Some locations, they might have the same tandem repeats, repeated like the same number of times. Other locations, they would be different. Okay. And we can figure out the total length of the SCR by multiplying the number of repeats by the number of nucleotides that are being repeated. So 3 nucleotides being repeated times 10 gives you 30. Okay. Now, since we know the location of these STRs in the genome, we can design primers to amplify them. Okay, so what we're going to do is we're going to take this and then we amplify. Amplified and clone means the same thing, like to like make many copies of it. That's basically what it means, right? Amplify these STRs using PCR. So now I have it in a tube. Okay, this tube contains a mixture of, like, the blue ones, you copy it many times, the green ones, as well as the red ones. Okay, like obviously you can't see them. They are like, invisible to the naked eye, and they're not even colored in real life, right? Okay, so same thing over here. Same thing for Sally. Okay, I'm just gonna copy it and then paste it over here. There we go. And we are going to load it on a gel. Okay, load it on a gel. So that's the well. I'm just gonna have a bunch of wells here. Now we don't have to load up all of them. Right? We can just, pick a lane and then load them. Okay. Load the sample into a gel. Remember, the gel is like this mishmash, right? You can't see it. It's like really smooth to our naked eye, but microscopically it has all those mishmash, right? Negative terminal here, positive terminal here. We're gonna run the current. All right, I'll give you a moment to draw all that. And then I'm gonna tell you what the results are. Okay, hopefully you're good. So let's take a look at Bob's sample. Bob's samples has 3 kinds of DNA. Those that are 30 base pair long, 6 base pair long, and 60 base pair long. Can someone please tell me which one is going to be closest to the well, closest to where you load the sample? You're going to load the sample here. You're going to separate it with a current. Which sample is going to be closest to the top? Is it the blue, the red, or the green? As in, after you run the current, which one is going to be closest to the top?
Blue.
The blue is going to be closest to the top. Why is that?
Because it's the longest.
Is it the largest though? This is 30, this is 6, this is 60. Right? Let's try again. One more time. Which one is it going to be closest?
Oh, 66.
60. That's right. The green one, right? The green
60.
one is the green one is the longest, right? 60 is the longest, right? So that's why it is going to be closest to the top. This is the 60 base pair. That's why you need to do this in color, right? So you can tell which band is which, right? And then which one is going to be the lowest on our gel? Right? The 60 band is gone now. Is blue going to be at the bottom, or is the red's going to be at the bottom?
Red, of course.
Of course, right? Because that's the smallest one. There you go. Right? And then blue is going to be somewhere in between, right? Like right here. Okay. I can't turn my video on. It is lagging. I think the program doesn't want to see me. But I'm doing all these gesture, and then I realize you can't see me. Anyway, so that's what it is. Okay, that's what it is. I hope that makes sense to you. All right, let's take a look at Sally. Okay, Sally. Which one is gonna be closest to the top for Sally? That's right, because the blue is the longest one, right? But it's not gonna be all the way up here, because now we have a reference, right? We have a reference to compare it to, right? Like this is the 30 in Bob's, so I should see the same band at the same location for Sally. Okay, we have the 20 next. Okay, 20 is going to be a little bit further down, compared to the 30, but not all the way down to over there, because that's like 6, right? If you get that far, you're 6. So 20 is going to be like around here, I would say. Again, like it's the relative position that matters, right? We can't predict the exact location, right? And then the 15 is very close to the red. Okay, I would say like right below it, because 20 and 15, it's super close. All right, so here you have a different banding patterns between Bob and Sally, right? With just 3 locations. Okay, so that's how you distinguish one person from another. So like, at the crime scene, if you got like a sample, right? Like a reference sample. Okay, and then the reference sample looks like this, right? With 3 bands. Then you know it's Sally. Okay, and it wasn't Bob. Okay, there's a question. When you have 2 different samples, how do you know which DNA is from? Because you prepared the sample, right? Like you would have to. That's actually a good question. Like I didn't really fully extend it. So like if you were really going to do this, what you need to do is you need to have a DNA sample from Bob. And when I say a DNA sample, I mean like you can swip his cheek, right? Like with one of those like kind of like those COVID swap stuff. You swap his cheeks and you collect his cell. So you have all of his DNA. But we're not gonna load all of his DNA. If you load all of his DNA, you're not gonna see anything. Okay. You have to amplify specific sections. So when you mix all of his DNA with the primers that you design, okay, so like for example, you design a primer that flanks this particular STR, and then you have another primer for like the red one, and then another primer for the green one, then only those part of his DNA is going to get amplified. Okay, so even though you originally start with all his DNA, but after the PCR process, you will only have 3 fragments of DNA that are in billions of the copies. The blue one, the red one, and the green one. And then it's all going to be in this tube. So when you load the sample into well number 2, like because you prepare that, right? So you know that has to come from Bob. Is that what you're asking me, or like am I totally missing your point here? I'm sorry. Could you like, okay, you get it, right? Have I answered your question? Or you have a follow-up question? Okay, all right. So that's only 3 STRs, right? But 3 is not enough. Like if we actually want to do this in real life, 3 is not enough, because people who are related probably have the same repeats in those locations. So you cannot, let's say there is 2 brothers, right? And you recover a sample. If you just look at 3 STR, you can't tell if it's the big brother or the small brother that was at the scene, right? Okay, so when we actually do DNA fingerprinting, you have to look at all the STR that we know of. So that's why when we look at this picture again, okay, look at it. Look at all these bands, right? Each of the band represent a particular STR, right? So you can see in this case, I am just guessing, okay? Like I'm not a forensic science person, but like this I'm guessing this would be like a sample from the crime scene, and then you have suspect 1, 2, 3, 4, 5, 6, 7, 8. Okay, eight suspects. Okay, each of them has very unique banding patterns, and you can see, look, this is a match, right? Look, I see a band here, I see a band here, I see a band here, I see a band here. Like it's like a perfect match. You understand what I mean? Right? So you don't just look at 3 locations. You look at all of them, right? I don't know how many locations this is. In a way, this STR creates a unique barcode, if you would, for a person, right? Like this is a unique barcode, right? And we're comparing the barcodes. Like if it's the same barcode, it's the same person, right? So even for people who are related, like mother and daughter, for example, they would have similar bandings, but never identical. Because the mom only gives the half of her genetic material to the kid, right? The other half comes from the father, right? So the banding pattern is going to be a mix of the mother and the father. So any questions about STR analysis? Okay. That's just showing you how to load the gel. That's just apparatus. And I forgot to talk about this slide, but I can tell you now. I keep on telling you, like the blue line is not the DNA, right? The blue line is just a dye that gives you an idea. If the blue line is here, your DNA samples, like the longer ones are probably up here, and then the shorter ones is here. So I need to stop the current. If I keep on doing it, all these will exit into the gel and you will lose them forever, right? So how do you see the DNA then, right? Well, it turns out you have to stain the gel. Afterwards, you literally, let me see if I can turn on the video again, since I'm not drawing. So you literally take the gel out, okay? And then you put the gel in a solution called ethidium bromide. Ethidium bromide. Okay, this is this like orangey color liquid that you would mix it in the buffer, and then you put your gel in there and you soak it there for like 20 minutes or something. So this thing looks like this, and it's gonna bind to the DNA and cause it to glow, to fluoresce when you shine a UV light at it. Okay, because the stuff binds to DNA, you don't want to be handling it without gloves. Okay, if it gets on your hand and then it gets in your body, it's carcinogenic. It says on the bottle it's carcinogenic, right? Because it binds to DNA. Right? It will alter your DNA. So you need to be very careful when handling that. And you can't just dump it down the sink because it's a hazard, right? And so what happens is you will stain this stuff and then you will shine UV light at it, and then you will see the gel. You will see the DNA, I mean, right? So these are the DNA that you have amplified with PCR, for example. Okay, and each band would tell you like how long the fragment is. Okay? You kind of leave behind traces of DNA sample. These little tracks that are left behind. Those are the fragments that we did not amplify. Okay, remember when we load the what was the guy's name again? Bob, right? When we load Bob's DNA, it was all of his DNA, right? But then we use PCR to amplify certain sections, right? So these tracks that are left behind, those are the section that we did not amplify. Okay, so this is the negative, this is the positive, this is where the wells are at the top, and these are the shorter fragments, and these are the longer ones. Okay, all these are same length though, I would say, right? But how do you know? How do you know this one is 268 and how do you know this one is 172? Actually, you can buy what's called a ladder. Okay, so this is a ladder. Okay, a ladder. You can buy it from these biotech companies. A ladder is a bunch of DNA fragments that are created to be certain lengths, right? So if you load it along with your sample, you can use them as reference. Right? So if you see a band here, you go ahead and you look at the ladder. It comes with a little pamphlet, right? I guess, and it tells you, oh, this one is 268. So if you see any other DNA that's around that area, it's going to be 268. Okay, so that's all. This is the STR, right? I'm not going to go through it again, but like there's a STR here, and then these people will have the same STR at the same location. What differs between one another is the length, right? So this person has a really, really long STR compared to the other one. That's why his band is really high up, right? And then the other one is really, really short. That's why it's all the way at the bottom. Okay. Not sure why these people are not happy though. I forgot the context of this picture. But there you go. Right? Just 2 bands. If you analyze all the STRs known, then you have multiple bands. Okay. Done. Okay, let's try this. Nobody got this right yesterday when I did it in class. Let's see if you guys will get it right. The plasmid is treated with ECOR R1 and BAMH when the digested plasmid, digested means after you cut it, right, is separated using gel electrophoresis. How many bands will you see? Okay, what do you think? How many bands will you see? So ECOR R1 and BAMH. So ECOR R1 will cut here. BAMH will cut here. So think of a rubber band. Now try this out yourself if you don't quite get it. You think of rubber band, you cut it 2 times, then you have 2 rubber bands, right? 2 fragments, right? So this is 1 fragment. This is the shorter one. And then this here, this is going to be the longer one. You run it on a gel, you will see 2 bands. Okay? You see 2 bands. There are 2 DNA samples with an STR polymorphism. Sample A contains CAG repeated 10 times, while sample B contains CAG repeated 15 times. Which sample will go further down the gel when separated using gel electrophoresis? Okay, this is CAG repeated 10 times. So that's 30 base pair in total. This is repeated 15 times, which is 45 base pair in total. I mean, you don't really have to figure out the total number of base pair. Like 15 is longer than 10, right? That works too. So which one is going to be further down? Well, it's going to be sample A because A is shorter. The shorter it is, the further down is going to be able to move down the gel. All right. Okay. We just have a couple other things we need to go through. Next one is RFLP. RFLP stands for restriction fragment length polymorphism. So a polymorphism is a difference. And this one is a difference in length, in the length of restriction fragment. What are restriction fragments? Restriction fragments are fragments, DNA fragments created by restriction enzyme. Okay. So let's say at the crime scene, there's some DNA was recovered, and let's say the DNA has 2 palindromic sequence here. Okay, CCGG, CCGG. So if I use a restriction enzyme that recognizes CCGG and I cut it, so one cut here, another cut here, then I would get 3 bands, right? Okay, 1 band, 2 band, 3 bands, and that's what shows up in the gel when I load the sample. Okay, not the original sample, but the sample that has been digested after you cut it and then I load it. I will see 3 bands. Now, because of things like neutral mutation that we've learned before, where you have a single nucleotide that has been mutated, is possible instead of CCGG, right? There is a mutation that causes it to become AT instead. So if you take a sample from someone with that mutation, and then you treat it with the same restriction enzyme, then now you will only be able to cut here and not here. So if you lower the sample, you will have only 2 bands, 1 band, 2 bands. The shorter one is at the bottom, of course, right? Which is the same size, by the way, like this one and this one. They would both show up here. Okay. But then for the suspect, there is only going to be 1 band up here. In fact, if you do the math, if you add up these 2 bands, the length of this one and this one is going to be the same length as that one, right? But the point is, if you analyze the restriction fragments, because so many of us have various silent mutation in our genome, the products that you get from digestion is going to be different for different people. So that's another way of doing DNA fingerprinting. You don't have to analyze the STR, right? You can do what's called RFLP analysis. Okay. So that's as much you know, as a DNA thing. Sorry, yeah, go ahead. There's a question. Yeah. Okay. So from the beginning, I suppose, or like, do you have a specific question? Okay, sure, no problem. Okay, you understand if you expose the DNA, if you expose the DNA to restriction enzyme, then the restriction enzyme will cut the DNA, right? But it's not gonna be cutting it like randomly. It's only gonna cut it if there are specific sequences, right? So in our example here, let's say the restriction enzyme will cut CCGG. So this DNA has 2 of such locations. Okay. In another DNA sample, it doesn't have to be the suspect's DNA. It's just like someone else's DNA. There's only 1 CCGG. Now you might ask, how come this one is not CCGG? Well, this person had a mutation that causes the C to mutate to an A. Right? And mutation can happen, right, during DNA replication and whatnot, right? So in your body, in my body, in someone else's body, we have a lot of these single nucleotide differences that are created from silent mutation. That's why where the restriction enzymes cut is going to be different for you and for me, because we might not have the same restriction enzyme sites at the same location. Like you might have both CCGG here and here, but for me, because I have the mutation, I only have 1 recognition site. Right? For someone else, maybe they also have another mutation here that caused this to become an A and a T. Then for that person, that DNA will not get cut at all by the restriction enzyme. Okay, so depending on whether or not there are mutations in the recognition site, that's going to cause the fragments, the number of fragments, to be different. Are you with me so far, or you want to ask me something about what I've said? Okay, so that's basically what it is. You run the sample, you expose the sample to restriction enzyme, and you want to see how many times it cuts it. Okay, no 2 individuals is going to produce the same cut if we look at a bunch of restriction enzyme sites. Okay, this is an oversimplification, right? We're looking at just one, like two locations. But if we consider the whole genome, then we're going to be able to create unique banding patterns. Okay. I don't think we're going to be able to finish today, but I'll go through as much as we can. Probably just a little bit more. Okay, so we're going to talk about gene therapy. Gene therapy. Gene therapy involves introducing genetic material into a person's cell. Okay. And you can do it to their bodily cells, which are somatic gene therapy. So somatic gene therapy will only affect the person who's receiving the treatment because we are introduced genetic material into cells that are not sperm or egg, that are not germ cells. These are cells that are not going to be becoming sperm and egg, I should say. And the effects of the gene therapy will only restrict it to the person and not future generations. There are also germline gene therapy where you are making modification to the germ cells, right? For the cells that will become the sperm and egg. In that case, it's not going to affect the individual receiving the gene therapy, but the future generation from that individual. So in terms of ethical concerns, germline gene therapy is a no-no. Okay, I don't think. I mean, I'm not sure if they're actually doing research on it. Maybe someone is, but then the ethical guidelines in general is that we should not be doing gene therapy because now you are affecting future generations. You are affecting someone who hasn't been born yet. So what gives you the right to change their DNA, right? But some people argue, hey, if we can modify the genes so that the person is going to be born with resistance to HIV, to AIDS, and that's possible. Right? Some people actually, I'll teach you this next semester, but some people actually have the genetic makeup to cause them to not be able to get HIV. Okay, they won't get AIDS at all. And we know what kind of mutation allows them to do that. So people are saying, hey, if we can engineer that into the sperm and the egg, then the kids are going to be born not having to worry about HIV. Isn't that a good thing? Well, right? That's the counter argument, right? But then, the counter argument of that is you won't know. You won't know the effects of the gene modification until the person is born. What if there is some unintended consequences that we're not aware of, right? And then the person is forever affected because of that. So, anyway, my point is somatic gene therapy, yes, there are ethical guidelines, but then for the most part, it's the area of active research. There are many successful stories of somatic gene therapy. Germline gene therapy, not so much. For now, anyways. Okay, so let's move on. The goal of gene therapy, what are we trying to do with it? Okay, we're going to talk about three different desired outcome, three different aims. Number one, replace a non-functional gene. That's probably the most obvious goal. I'll give you examples of it later. You can also be suppressing an overly active gene, or you can use gene therapy to modify the clinical course of a condition. Let's look at each one of these more closely. First, if you have a cell with a non-functioning gene, okay, what's the logical thing to do? Well, you give them the gene that's functioning. Right? For example, sickle cell anemia. You remember that, right? We talked about this multiple times. That's caused by a mutated hemoglobin gene, and you are not making functional hemoglobins. Okay? So the logical thing to do is to deliver a functional copy of the hemoglobin gene, and then the cell will start functioning normally again, right? But these type of conditions usually have to be recessive. Right? So for example, if you're little h, little h, right? And this gives you the sickle cell anemia. I can provide a functional copy, capital H, and then it will override It will override the non-functioning copy, and now you will be okay. However, if the condition is dominant, okay, which means the disease allele is actually capital H, and I try to deliver the functional one, which is like another H, is not going to work. Because the dominant gene, as long as it's there, it's going to override whatever functional gene you introduce. Because the functional gene is recessive in this case, it will cover it up. It will get covered up by the big H. It doesn't matter how many normal copies you deliver, the big H is going to suppress all of them, and it's not going to work. Okay, so for replacing a non-functional gene, typically these conditions has to be recessive. Okay, if it's a dominant situation, you have to find a way to knock out the dominant copy before you deliver the functional copy. Okay, so this is only successful if the effects of the disease are reversible or have not resulted in lasting damage of the bodies. Things like sickle cell anemia. Beta thalassemia, I told you before, that is caused by a nonsense mutation where a whole chunk of hemoglobin is missing. Hemophilia is a bleeding condition. People are missing what's called like a clotting factor for your blood to stop, for you to stop bleeding. There are a bunch of clotting factors that have to be working together. So if you're missing one or more of those, then your blood don't clot quickly. Okay, like even if your nose bleeds, sometimes you have to go to the hospital to stop that. And in the case of gene therapy, you would introduce the missing clotting factor, the gene that makes them anyway, and then you will fix the problem. But if the condition causes some damage to the body already, okay, let's say there is a mutation that causes that interferes with mental development. Okay, so after let's say at age 7, the person received the gene therapy to fix that. All the damage that was done, for the 7 years of the life is not going to be fixed. Those are irreversible damage. Okay, you cannot gain back the missing cognitive development that was happening in the 7th year, right? So some diseases is not ideal for this kind of gene therapy. Okay, it's only for situations where you haven't done too many lasting damage to the body, and as soon as you give them the functional copy, things are going to be okay forward. All right, second goal is to suppress a gene. Sometimes a mutation causes a gene to be overly active. You might remember gain of function mutation, right? An oncogene, for example, right? The green light that we talk about, right? When an oncogene is created, the cell keeps on dividing and dividing and dividing. So in this case, we want to create a gene that will go into the cell, and then it will suppress the oncogene, right? If we're able to suppress the oncogene, then the tumor will not grow anymore, right? It loses that green light. It loses the signal to keep on dividing, right? So that could be used in conjunction with, say, a surgery, right? You remove the tumor as much as you can with surgery, and then you do the gene therapy to suppress further growth, and then you can use, say, chemotherapy to kill off whatever remaining cancer cell is in the body, theoretically. Right? Next, we have the third goal, which is to modify the clinical course of a condition. Right? So maybe we can, in this case, instead of suppressing a gene, we can introduce a gene that would trigger cell death. Okay, or we could insert a gene that causes the cell to produce certain special markers on the cell membrane. And when the immune system see those markers, they will attack them. Right? So that's ideal for cancer again. If you're able to insert a suicide gene, that's what they're called, into the cancer cells, the cancer cell will kill themselves. Okay. If you're able to insert a gene into the cancer cells and cause them to create these essentially like glowing signals on the cell membrane, the immune system will have no problem finding them and destroying all the cancer cells, right? So here it is. This is a disease cell. Doesn't have to be cancer. It could be like something else. But we introduce a suicide gene. The suicide gene contains instruction to make a toxic product, and then it kills the cell. But of course, we have to be very careful. We have to make sure that only the cancer cell is going to be receiving this suicide gene. We don't want this suicide gene to be going into healthy cells. That would be like terrible outcome, right? You can create a gene marker, and now we have these like glowing signals on the cell membrane. Immune cells will recognize them, tag them, kill them. That's the theory. Two more slides, and then we're gonna stop. One more slide, we're gonna stop. Okay. So the gene therapy can happen in vivo or ex vivo. Ex vivo means we have to take the cell out of the person's body. Okay? For example, bone marrow cells, we can take them out. Bone marrow cells contains cells that will become white cells, which are really useful to fight off diseases. And then we genetically modify them with the genes that we want, and then we put them back into the person. Right? So all the gene therapy happens in a petri dish. Right? In a way, this is better to avoid off target. Okay? You want the gene to go into certain cell type. So if you're modifying in a dish and then you're giving it back, you know it's not going to go into other cells. Okay, but that's not possible for like, for like the suicide gene that we talk about, right? We cannot take out the cancer cell and then put the suicide gene in and then put the cancer back in. Like logically, that doesn't even make sense. If I'm able to extract the cancer cell and put it out, then like we are done. We don't even have to put it back in, right? The problem is like we don't even know where the cancer cells are. So in that case, we have to do in vivo. In vivo is, inject the genetic material. We use a vector to deliver the DNA, and hopefully the DNA is going to go to only the cells that we want, and then only affect the cells that we want to affect. Okay, so ex vivo, you take the cells out, you do some genetic modification, you put them back in. That's useful for, let me give you another example. You can actually take out the white cells, okay, and you can genetically modify them to kind of educate them, to teach them, and say, cancer looks like this. Okay, now I'm going to put you back in the body. Go find them and kill them. That's an example of ex vivo. Okay, in vivo, everything has to be happening inside the body. You just inject the DNA with the vector, and let the body does the rest. Okay, that's it for today. We just have a few more slides to go through next class, and we will do lecture 10, which is the last part of the course. Okay, you only have a 20%. Oh no, that's not true. You also have your quiz. Yeah, your quiz is gonna be next Tuesday, and I'll post the information for it later. Okay, that's it. Thank you for coming. Enjoy the rest of your day and week. Bye for now.